Hi there.
Welcome to Microbiology Journal Club. We nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology. Then I was a fact checker and editor for pharmaceutical advertisers. Nowadays, I'm a member and the president of Biotech Without Borders, a nonprofit based at NYC, dedicated aiming at the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked in bacteria, but I've also worked as a research integrity specialist and currently working as an editor for a scientific journal.
We've been off for a month, but typically every other week we meet to talk about microbiology. Like today, we do an overview of some of the coolest microbiology papers that we've seen in the last 2 weeks.
Yeah, and we also try to select a paper to do deep dives on. So usually if it's recommended to us, so please message us at MicroDWC, at Mastodon, or at Twitter, or wherever you can find us, and let us know if there's something you've read that you really want us to take a deep dive into.
Absolutely. You can follow along with the papers that we discuss in our shared Zotero library, our social media handles, and the link to that library are in the doobly-doo below.
Yeah, so we really want to hear from you. So please use comments or or tweet us at MicroTWJC. Yeah, what kind of papers do we have today, Danny?
Right. So the things I've come across in this past little while is some microscopy of bacteria stuck to the sugar and silica encrusted over microalgae, looking at SARS-CoV-2, defective viral genomes, looking at these abundant free-living cyanobacteria that can also metabolize chitin, that's the stuff in like arthropod shells, looking at streptomyces spores that hitchhike on modal bacteria, and influenza as a model of liquid biomolecular condensates, which I believe is very hot right now.
Yeah, I think they're very hot right now. Not only that, we have can we cure HIV using gene therapy? Can we fight antibiotic resistance by targeting the resistance genes directly? And we see how phages can puncture in between two membranes at the same time. We also see how to program a protein delivery system to do what we want. And we also can you see how people can use computation to create biosensors and engineer and how they work. Um, so yeah, thank you all very much for joining us. We'll bring us to the first paper, which is
Yes.
let me cue it up quickly.
Generation and functional analysis of defective viral genomes during SARS-CoV-2 infection. Yeah, so anyways, if folks haven't joined us before, like we really started during the height of the pandemic lockdowns, and we used to talk a ton about just like trying to keep up with all the SARS-CoV-2 literature. But actually, in the last couple episodes, we really haven't touched on SARS-CoV-2 at all. I mean, at some level, the publication frequency for this particular topic has slowed down.
Mm.
But also, a lot of the stuff that's coming out, like, I don't know, it wasn't it's kind of tiring to talk about it over and over again. But I chose this paper this week because I'd never heard of defective viral genomes, and it seemed like a kind of interesting way to learn about this concept, like maybe notify some folks about it, but also like return to this idea of SARS-CoV-2 because I mean it's still circulating. I don't know what it is like in the UK, but like the wastewater treatment stuff in the US like has had a spike recently. I mean, we don't have any official uh, surveillance anymore, really, for it. But yeah, so it's still
Yeah,
sort of circulating around, and I think understanding some of these mechanisms and also just keeping it in the public discourse is sort of a good thing.
yeah, because since we're not doing surveillance anymore, there can't possibly be any. I can't see any SARS-CoV-2, therefore there can't be any SARS-CoV-2, right? That's how it works.
Yeah, apparently. So
Yeah.
this one dives into this concept of defective viral genomes. So apparently, it's been described in other viruses before. So in influenza, I think it's been discovered. And what it means is that there are some genomes that, for one reason or another, when the polymerase is making it, it sort of like drops off and then reconnects at another point. So it's missing. Yeah, there's like a gap that gets missing in the sequence of these genomes. And it's been shown to actually have in influenza, like defective viral genomes, like have a different interferon response pattern that gets shown. So there's some thought around the idea that creating these genomes does modulate pathogenesis in some way. Again, pathogenesis is like the super complicated thing where it's a little bit of the host and a little bit of the pathogen. So it's unclear like who it might benefit to have these defective viral genomes. But the point of this paper is to figure out what are they? Do they exist in SARS-CoV-2? Because people do think that they do at some level. What you might see is like this is kind of similar to recombination in some ways, right? Like sort of like an accident of the polymerase that accidentally creates some other variant of some sort. But I don't think people don't think of these defective viral genomes as being infectious of themselves. It's more like part of the population of viruses that sort of inhabits somebody. And the way that they're studying it in this paper is they go in with a bunch of bioinformatics. They're looking at data sets of SARS-CoV-2 that's being sequenced in a bunch of different scenarios, and trying to use some software to figure out, find these junctures where the polymerase skipped a bunch of stuff.
Yeah. So I've heard about like DVGs, and more like in the context of like artificially created. So like the concept of like for certain gene therapies, because viruses have only so much they can fit into their shells, cutting out important genes for the virus, and then having a helper virus help this defective virus to replicate.
I've heard of that as well. That's like a way of trying to make sure that you're not propagating these like gene therapies in a way that they're replicating everywhere. They can only replicate in the subset of things that they get some helper molecules at.
And also, they're like, I think virophages are like some variant of this, right? Where you get a virus that kind of that only can infect cells when another virus is present, and so it kind of parasitizes them. Usually, they're a defective form of that virus. But thing is, those concepts, I always thought of that as a unique concept. But from this paper, it seems like these defective viral genomes are a different variant of that, where they're just there as a background for all viruses. All viruses acquire mutations, and so they produce these as part of their infection. And this is just pointedly looking at what is happening to generate these DVGs.
Yeah, like to me, the way that the paper seemed to present it is that it's about the diversity of different things that could just be during a single infection. And we've talked about that before, like because so much thought has gone into SARS-CoV-2's evolution, right? Like how does it acquire variation and this sort of stuff? So this may also be one of those ways, like it creates like some variation in the infection. Maybe those little defective viral variants are good for recombination in different ways. I don't know. But the paper itself is not really concerned about all these questions that we've just like posed, but mostly just concerned about describing, let's look at these defective viral genomes inside of SARS-CoV-2. So as I said, they do it bioinformatically, and one of the first things that they find, in this figure here, I think the C, they like see the breakpoints are in like a very specific area. So it's not like you can get defective viral genomes where the polymerase skips all sorts of areas. They actually typically stop replicating at one point and skip to another point, which sort of helps the hypothesis that there's something intentional here, right? In like evolution, that like you might want to have some of these defective viral genomes floating around. Like SARS-CoV-2 has some mechanism maybe to direct where they will appear, like what the defective genomes might look like.
And an important thing I always had to remind myself of this is SARS-CoV-2, this polymerase has proofreading activity. So compared to the other viruses mentioned, we should expect a lot more stability and less of these. But the fact that there is one specific point indicates that there is something interesting going on there. That something is specifically like the polymerase just can't deal with these specific issues, and it's causing that to.
Yeah, totally. And they hypothesized there that like secondary structure could be some sort of part of that. And so they go in and they try to do some analysis on, does this correlate with secondary structures that they see? And what else do they do in this paper? Oh, and then as I was saying before, they end up, it's been hypothesized or maybe shown in influenza that those defective viral genomes do elicit a different immune response. And it makes sense, right? Because if you skip a bunch, you might have a different you might not make so much of a certain protein. You might look like a different piece of objects. Like this stuff is, it's like it's an interesting thought that like just by skipping a bit of the genome, you might have a different immune response. Anyways, they follow up with that to try to actually test out some of these defective viral genomes and whether they elicited a certain immune response in people.
Yeah, fascinating.
I don't really know what else I can say in depth about this, like in terms of takeaways. Just that they do exist. They do find other patterns, but again, I didn't read this deeply enough to sort of share with you guys what I think those patterns are that they pulled out. Because like they do talk about. I just want to say, like I did read, like they did talk about seeing like that variation inside of like an immunocompromised individual. I think they look into an immunocompromised individual and they say, oh look, like with these defective viral genomes, like in an immunocompromised position, you see a whole bunch of them. So something about like the diversity of it. They try to correlate that with age and severity as well. They have this metric J frequency that they talk a lot about in the discussion, and because they are trying to ultimately find some sort of correlation with phenotypes, and this is one of the metrics that they're using.
Yeah, I mean, it's interesting. I mean, I'm just curious about like what that means for testing as well, because if these defective virus particles are there, what does that mean for when you're testing for SARS-CoV-2? Does that affect how good tests are or not?
Yeah, like I think from what I was reading, there's not a high frequency of these defective viral genomes in any individual infection. So like they don't make like a huge proportion of things. But actually this figure that you're on right now, figure 7, that it's interesting, right? Because this is part of the trying to correlate it with phenotypes. And of course, like a big phenotype that we know for SARS-CoV-2 infection is asymptomatic versus symptomatic infection. And so like one of the correlations that they do find is that total defective viral genomes and unique viral genomes, they're higher in symptomatic infection, right? Which again is trying to give us this hypothesis or thought that they're playing some role in the productive infection and the life cycle of SARS-CoV-2.
Yeah, I mean, it's a difficult hypothesis to put forward because someone could always say that, hey, it's because symptomatic viruses just have more virus, so there's more chance that these will be produced. And I think they try to address that by trying to like adjust for it. If I believe that's what's going on in Figure 7C.
Yeah, I think they are. I think they are, but I'm not 100% sure. Yeah, and it's an interesting concept. Something that reminds me of when people talk about like, so RNA viruses, first of all, they always say like all those copies of it, like they're gonna be they're gonna have variation, right? Because RNA, but you said proofreading activity means that maybe not as much variation. This is just another way that variation can embed itself into RNA viruses that replicate.
Yeah, completely. So that's
All right.
Yeah,
that's all I want to say about that. So moving on to our next selection.
Yeah, CRISPR editing of CCR5 and HIV1 facilitates viral elimination in antiretroviral drug-suppressed virus-infected humanized mice. So the big thing is that, this is in mice, so let's not get too excited because mice don't naturally get HIV.
So I think they even had to humanize the mice in this model to make sure that they could get HIV to have an infection model.
This is very much a proof of concept in an organism that might not necessarily reflect what will happen in like humans or other primates. But it is interesting just looking at the strategy they use, because what they are doing is there are 2 layers of what they're doing. So first thing is that they're trying to hit latent HIV. So HIV as a virus, when it attacks, one thing it does is it can actually intertwine itself in your genome, in the DNA in your nucleus. And then, so even if you destroy all the viruses in that cell, it's still in your nucleus and it'll still replicate after that. So one of the things that they were looking at was doing a CRISPR treatment which can hit the viruses that are integrated within the genome.
Yeah.
So I think they did that in previous research as a proof of concept,
Mm-hmm.
and they found that they could pop out those viruses. They could
I think we've seen I think we looked at hepatitis or something. People have been investigating this idea of CRISPR going in and excising integrated genomes of viruses. So yeah, it's like they're really, I feel like this paper is sort of taking this stuff that they've done before, showing that it worked, right, in some limited way, and putting it together in almost like a therapy style. Like, so then what does it mean to like use this in concert with state-of-the-art antiretroviral therapy, and maybe together these two components, because antiretroviral therapy is like reducing the viral load, how much virus is actually replicating. And then the idea is that then they add the CRISPR section, and now you're also targeting these latent viruses that are recombined into the cells.
Yeah, I think that's important. Hitting the virus at multiple like points is kind of the main like core of HIV treatment these days. And the thing that they add on to this is CCR5. So CCR5, if you're not aware, is the surface is a very important surface protein for HIV to latch onto cells. And I think it was discovered about like maybe 10 or 15 years, a long time ago, that there are certain people who are naturally resistant to HIV because they have mutations in the CCR5 gene. And part of that is why there's certain people, I think maybe 2 years ago, who got like cell transplants, who got bone marrow transplants from with cells that had that mutation that led to people being cured of HIV.
Yeah, it's like missing some part of the CCR5 gene, and that's a part that like HIV binds to. But I think HIV does have some diversity in that respect, though, right? That they're not all totally dependent on CCR5 in order to enter, and so so it's not a silver bullet in the strictest sense, especially.
I think that HIV evolves fast enough that I think there are other surface molecules that HIV can use, but I think people have been focused on CCR5 because of the big findings.
And I do want to say too that this is a very interesting target, because like I don't know if folks have heard, like maybe a couple years ago, that there was like this scientist who was denounced by the community that created these twins that had CCR, like twin human beings that had CCR5 deletions. And like that was quite controversial in the scientific world, because obviously like we don't condone this idea of like human experimentation, and like the fact that we're seeing, like no one did this mouse study at that point, right? And they went ahead and sort of forged on that. So anyways, this is it's almost like that concept, the concept of CCR5 has like as being like a good gene therapy to give humans resistance, like that's been like I guess a dream for some period now.
Yeah.
But it's important to realize there's a lot of like steps that have to be done along the way before you could deploy that sort of widely.
Yeah, I get the like kind of. I mean, for that particular controversy, it's like coming out of nowhere and saying that I've genetically engineered two babies out of nowhere is already going to get people's backs up. And the idea of like, and because this is a gene therapy, to genetically engineer these mice, like as they're being infected, to be resistant to HIV. And I'm sure there will be some controversy about that, that if it was actually moved into humans. So I think there
was no understanding of like the risk and stuff, because these studies hadn't been done. So I think what we're seeing now is like, okay, I guess people still are intrigued by this idea of CCR5 as some sort of resistance, genetic resistance that we could engineer into people. So going back and doing this work that really is critical before moving further.
And so here they're basically like trying to lock off like as much of the HIV's life cycle as possible. So targeting the latent versions, using antiretroviral therapy to stop it from replicating, and targeting CCR5 to stop it from moving between cells. And then hopefully using that to break its life cycle enough that it stops replicating entirely. So they do a number of measures. So you can see like they got the CD4 cell count. So the CD4 cells are the T helper cells, which are usually the target of HIV. And then they look at the number of mice that have plasma viral copies. And I think what they find is that, like, although their treatment group, I think there are only 4 mice that managed to keep their HIV infection. So throughout this study, you'll see there's lots of dots at the bottom, which are like below detection, and then maybe 4 dots that are the mice that managed to still stay infected even after all of these treatments. So
yeah, I mean, one thing I'll say is that art, it's also hilarious. It's called laser art. I guess that's like in reference to the specific drugs, not in reference to light or anything like that. But like art, antiretroviral therapy is super effective. Like there's been so much work to reduce it, and a lot of people are saying that like if you go on antiretroviral therapies, like when you become undetectable, it's also untransmissible, the virus. But the issue would be there is that I guess you're going to be on those antiretrovirals for the rest of your life. So I think that like the sort of therapeutic area that they're hoping that this targets is that, yeah, maybe if you can't take those antiretrovirals for the rest of your life, there's probably like liver issues and things like having so much. Like maybe this gene therapy would be helpful there. Yeah. So, anyways, there is use. I guess what I'm trying to say is there is use for this gene therapy, even though that we have very effective antiretroviral therapies. But at the same time, like we don't really know like the risk benefit analysis. Yeah.
I think it's also the fact that there's infrastructure that, so in the Western world, we can get these retroviral therapies very quickly to people, but lots of people are infected in places like South Africa or other places where there are big like problems with HIV. It's the infrastructure isn't there to keep those continuous treatments up. So having a one and done therapy would be really useful in those situations.
Yeah, absolutely. That makes a lot of sense.
Yeah. And yeah, this is just like an amazing study because they do like 2 subsequent like gene therapy steps plus all these other treatments to tackle HIV. And it is like, I mean, it's not fully successful. I mean, even in mice which are already slightly resistant to HIV, there are some mice that still manage to keep their infections. So there is a lot to go, but I think this is, I mean, quite interesting because they use like they deliver their gene therapies using adenoviruses, and one thing I was like interested in with this paper is they try to look at like where the recurrence is coming from. So once they eliminate HIV from everywhere, where does it start popping back up from? Because that can tell you about where your gene therapy is going and how well it's working. So
they very systematically go through a bunch of tissues to be like, is it still here? Is it still here?
Yeah, I think that, and that's a very important thing and very, very interesting, especially from the perspective of anybody working on a gene therapy, because you want to understand where your vehicle's going, and where it's not going, so you can design better delivery systems in the future, so you can get better coverage of cells. So yeah, I think that.
Yeah, I mean, I think that's also important. Like, yeah, it's just highlighting like where the state of our technology is and where it could be is actually like quite much further, right? Like targeting is its own very unique and difficult set of challenges. And yeah, like we sort of haven't even breached that with this particular study.
Yeah. But even so, there's some big things like you have a CRISPR therapy that can legitimately change mammalian cells in multiple ways, be delivered multiple times without there being immune response against whatever is delivering it to make it less effective. There's a lot of like different steps in here that are very impressive and things that could be good for the future.
Yeah, absolutely. That's all I want to say about that. Are you good?
Yeah, I'm good. And speaking of CRISPR, we're looking at another paper called Removal of AMR Plasmids Using Mobile Broad-Host Range CRISPR-Cas9 Delivery Tool. So antimicrobial resistance plasmids are a real bane because they hang out everywhere. So even if you annihilate bacteria, the plasmids can still transfer between different species. And in a sense, this whole antibiotic resistance crisis, we are fighting against genes as well as the bacteria.
Yeah, because we've talked about mobile genetic elements before, conjugation, like Graham well, actually, all bacteria, but specifically, I think people think gram negatives have all these really crazy ways of transferring genes between each other through plasmids. And so, yeah, like I think what you said, Fuzz, it's interesting to think about the fight against antimicrobial resistance not as the fight against very specific organisms, but also just the fight against these genes that are flowing between populations.
Yeah. And so that's based on that concept. I think the idea of using a CRISPR tool to attack the genes directly, I think it's an interesting one. And based on what we saw in the previous paper, it could be quite a nice route to explore.
Mm-hmm.
So
and I
yeah,
guess, oh yeah, go ahead.
sorry, no, you go for it.
Oh no, I was just gonna say. So I guess like the thing is that you have to get that CRISPR into all these different types of bacteria that could potentially have have an antimicrobial resistance gene being harbored, and so you need a platform that could get into all those different types of bacteria. And so the authors of this paper are there's I guess they found this plasmid. I looked it up a little. It was isolated from soil, and when they found it, they're like, oh wow, it's really broad host range because every bacteria has its own replication machinery and stuff. So not all plasmids, not all things could just replicate in each one. They are using a very specific plasmid that, for one reason or another, I mean, I think that's still a mystery in some ways. Maybe in the literature the answer exists, but I didn't dig in. But yeah, like for one reason or another, this plasmid like just replicates in a lot of different types of bacteria. So like this is a great scaffold. The authors are thinking this is a great scaffold to put our like antimicrobial resistance CRISPR sensing machinery. And because if this plasmid could sort of immunize, you could even think of it as immunizing like the communities of bacteria that can pick up this plasmid, then they'll no longer be able to hold a plasmid that has antimicrobial resistance on it.
Yeah, so they transform these into, I think, bacteria that have plasmids, and the Cas9 is targeted to the plasmids using the guide RNA.
Mm-hmm.
And the interesting thing is, I think they also do conjugation experiments, right, where they
Mm-hmm.
incubate bacteria which have this plasmid along with bacteria that have the antimicrobial plasmid, and they exchange genes. And we can see that, and they can see that once the bacteria get conjugated with, those bacteria will get their antimicrobial resistance genes eliminated. So it's almost like using bacterial mating as a delivery system.
Yeah, I mean, just like how the antimicrobial genes are being passed around bacteria, the hope is that maybe they would also pass around this plasmid. And I think that that's,
Yeah.
to me, the major question about this sort of technology is what incentive do the bacteria have to keep around this antimicrobial resistance CRISPR plasmid? Like, it is broad host range. It can go into all these different bacteria, but why does it want to keep it around? And I think they sort of talk a little bit about that in discussion. Like, there's still sort of an open question of fitness cost, right? Is this like too much of a fitness cost for bacteria to have around? And I think also part of it would be like, where would you use this? Where would you use this? Is it like proactively we're spreading it in hospitals or something like that? Yeah, but the concept is very great. It's really interesting, and I'm curious to see where it goes.
Yeah, I mean, there is like an evolutionary incentive for bacteria to not take up this plasmid. So I guess
Like
in that
not
case,
maintain it. Like maybe they'll take it up
yeah,
because they mated with somebody that had it, but like would they keep it? Because like what's keeping it around? Typically, when we think of plasmid maintenance, it's like there's an evolutionary advantage to having it. That's why antimicrobial resistance plasmids like they keep them because there's selection and they like the population of bacteria that has the resistance, they're surviving, so it's giving them an advantage. So yeah, like it's kind of interesting.
Yeah. So I'm not sure like how this will be practically used, but I do think it's an interesting concept.
Yeah, and just like the thought process of like that there are some things that like transmit through bacteria, right? Like there is a way of getting a wide range of bacteria to pick up some genetic elements. Like thinking maybe more about targeting antimicrobial resistance, as you were saying at the very beginning of this, not as like a very specific bacteria we want to get rid of, but rather some like group behavior of bacteria that we're trying to modulate.
True. I mean, you can actually target these to, like, say, stop virulence genes from propagating, or stop like other things that. So, because there are lots of like commensal bacteria that become virulent, and if you could you have bacteria like plasmids like this to essentially stop like modulate those communities so they don't become virulent, then that could be another useful thing to have.
Yeah, I'm curious whether or not that makes, yeah, if that's a viable strategy in some way. It would be really interesting to see something like that.
Yeah, like if you could have like one that's like, oh, a a bacterial plasmid that cuts out pentanvalent leukocidin, but still
Mm-hmm.
leaves the bacteria alive.
Mm-hmm.
That would still that'd be quite useful to see whether that can, like, whether you could get that to propagate through communities, and then
Yeah.
you've got like essentially turning a virulent bacteria into a probiotic.
Mm-hmm.
So
Mm-hmm.
I
Yeah,
think
like
that's
I wonder, like it actually really makes me think about like maybe we should be thinking about selection uh, selection pressures that we could put on bacteria in order to take these up, but that aren't harmful to us. Like kind of replacing one selection criteria, the antimicrobial resistance, with another selection criteria of some sort. I don't know what that would be like, but
yeah.
yeah, it's a strange thought.
Yeah, I mean, I think thinking about this as a native like gene therapy that bacteria could administer to each other is an like, I think that's another takeaway from this paper. So
Uh-huh.
rather than thinking specifically about the outcome, thinking about the methods that they use,
Mm-hmm.
those methods have like broader applications.
Oh,
Um,
absolutely, absolutely. All right.
so yeah.
So
Now this is an interesting one.
moving on. Oh yeah, this is our hot paper topic of the scientific
Yeah.
field. Defining basic rules for hardening influenza A virus liquid condensates. So yeah, I don't know how this trend, research trends, they're real. Like people get obsessed about certain topics and they become like very interesting, mainly because they become so explanatory. They're able to explain so many phenomena. It's like, wow, like
Yeah.
why didn't we study these things before? And so the topic we're talking about here is these biomolecular liquid condensates. So if folks know their high school cell biology, we're just, cells are bags of proteins and stuff all mixed together. The cytosol is just this dissolved like liquid that has everything that the cell needs inside of it. And it's only eukaryotes that have compartments that like allow them to segregate all these biochemical functions. Well, it's not true. Like what people have observed inside of the cytoplasm is, for various reasons, specific molecules can be crowded together to create these biomolecular liquid condensates, regions of sort of concentrated factors of one type or another. And so those regions can do very specific biochemistry that you can't just do if you're all diffuse in the cytoplasm. And specifically, this is quite interesting through the lens of viral infection, because we know viruses have to concentrate all their viral factors in a certain area to get stuff done. And so in this paper, they're thinking of influenza as a model for studying biomolecular liquid condensates. I don't even know if they think about influenza as an infectious agent in this paper so much. Like it's more of the physics of it that they're concerned about. Like they know that influenza makes these condensates, and they can see them in cells, and they just want to play with the parameters of the cells. Essentially, the cell is a petri dish, and they're able to instigate these condensates by influenza infection. Then they can vary things like the temperature. They vary the concentration of different viral factors. They add like a drug of some sort that like sort of helps bind the condensates together, like sort of instigates their formation. It's been shown in previous studies, and they're playing with these variables to try to get a sense of what's the behavior of these biomolecular liquid condensates. We need a short form for those. Liquid condensates.
Yeah,
what do people call them?
so I've heard them called like membraneless organelles is another one that I've heard.
Organelles. Yeah, I like
Because
that.
we look at the cell, we see lots of membranes. Oh, those are organelles, but these are things that we can't see because they're all made of protein or made of like a specific or specific molecule that binds together.
Mm-hmm.
So,
Mm-hmm.
so membranous organelles is one thing that I've heard people talk about, but I think condensates tends to be the one that people are focusing in on.
Yeah, we'll just say condensates, not biomolecular liquid, just condensates.
Yeah.
Yeah.
Everything we're looking at, everything's biomolecular if you're looking at a cell.
That's true. That's true.
Yeah. I hope no one will get confused if we. Yeah.
But I think you just brought up an interesting point that like typically when we look at cells and we're trying to find like the organization within the cytoplasm, we're using the membranes. We stain for some membrane thing so we can see the separations. But in this case, we are usually staining for proteins that we already know are in high concentration in these condensates. And that's the way that we're finding. So in this case, I think they use like the nucleoprotein, right, of influenza as the way. So like, right, like they're saying influenza, it needs to do influenza stuff in influenza zones. And so it's going to have a high concentration of nucleoprotein. And that's how they're going to visualize them in these cell images. And what I thought is interesting is they want to look at this phenomena of hardening. And so like their proxy, or the way that they sort of say, like, is this thing that they're looking at? Oh, yeah. All right. See you, Christoph. Like one of the ways that they're trying to look at hardening is that they're looking at do they fuse with other condensates, right? Or if they bleach a certain area, does that diffuse again, right? Can they bleach? Like if things are diffusing, if they're liquid, right, then you would imagine that if you bleached a section of it, it would all diffuse and become equal again. But if it's harder and not as liquid, let's say, then if you bleach a section, it won't return. And so I think that's like there's a very interesting set of methodologies in this paper that they're trying to get at, like, how do you even visualize these things so that they can, again, vary the parameters and see how it behaves under these different conditions.
Yeah. I mean, it's very because this is such a new field that they are trying their best to find out new ways to measure it, and I think this is an important part of this paper where they are applying like they know what's happening on a certain level, but they need to apply their models to define ways to observe these effects, and find just even like basic things like finding out like the kind of things that are going on. I mean, so I think these sets of like diagrams are describing what we're talking about, where like there are some proteins that bleach and then they move around the place, but then some proteins that bleach but they stay in one place. And I mean, I feel like the way that they establish stability is essentially by introducing a drug that causes like connections causes cross-linking between certain nuclear proteins. Which and then there's a fair bit of like, and they one thing they do is they heat things up and find that the drug makes things more stable. And I think there is an idea that perhaps that in future, once we understand these things more, we'd be able to actually derive treatments that can attack them. And so a new way to attack viruses is a potential.
Yeah, I mean, that's their ultimate goal, I think. But I mean, for me, when I read the discussion of this paper, like, it sounds like they're very concerned about this biomechanics of, right, like the biophysics of these condensates. And what they said was that there's no purely in vitro way of making them right now. Like like in vitro meaning like sort of this is in vitro because it's in cells or whatever, but like they're saying there's no way, like cell-free way to make these at the moment and really get an understanding of some of the physics behind it. So this is like the best model they have of just inducing condensates within influenza infected cells. And again, they have, as you said, they have a drug that also helps them sort of like adjust the properties of the condensates within the cell.
I have to say, for a paper, this paper had the most interesting limitation section I've read for a while. They do delve into like the limitation of the model, which kind of speaks to how much more there is to discover in this field and how to,
absolutely.
And I thought
Yeah, like I'll say another part that really interests me in this paper is that they want to measure Gibbs-free energy as a way of saying like, oh, like stuff that's diffusing into these zones versus out of these zones. And the way that they have to do that is that they're looking at the fluorescence of the zone of the condensate compared to the background fluorescence across the entire cytoplasm, excluding the condensates. Right, and then they do they do a little calculation to give like, oh, this is a diffusion rate, right, that they could calculate behind that. And I found that fascinating too, because it's like, that means the background staining that they're getting is being quantified, right, and plays into the way that they have one of these measurements. Yeah, and anyways, maybe I just don't do enough. I haven't done enough confocal microscopy sort of like in this interplay with the quantitative aspect of things. But yeah, like that's fascinating to me that this is one of the ways that we're going to come to study this phenomena that ostensibly is probably pretty important in a lot of places, because while this is happening in the model of influenza here, it's also happening in regular cell biology where cells are just doing things for themselves. And like, again, we've talked about, everyone knows, I think people know it, organelles, things that are membrane bound. And I'm always super excited about the stuff in bacteria, the protein bound, right, compartments. So another set of compartments that actually just seems so hard to study, but potentially just have a lot of promise for understanding the mechanisms behind why things go in cells.
Yeah. So I've said my piece on this paper. So
moving on.
So next paper is Cryo-Electron Microscopy of F1 Filamentous Phage reveals insights into viral infection and assembly. So F1 phages are kind of important for biotechnology. I think that they're part of a widely dispersed group of filamentous viruses, and they're kind of like, they've been known for a long time. And the thing is, they infect gram-negative bacteria, but the actual way they cause that infection has been somewhat very difficult to figure out. Partly because, and now we live in the era of cryo-EM, there could be a potential to find out like what is going on. How do these actually cause infections? The thing about cryo-EM though is there is like a size limitation on these things. So these are very long filamentous viruses. But what they've done here is that they've updated them. They've generated these nanorod versions of filamentous viruses. They figured out that, I think that the size of these viruses, it's dependent on the size of the DNA that they're packing up. So if they trick these viruses into packaging a much smaller amount of DNA, they become small enough to be resolved by cryo-EM. And then once you have cryo-EM, you produce a lot of these different viruses, and then you take pictures of them in all sorts of different orientations, and then the clever computer algorithm puts it all together to develop a 3D image of it. And
yeah, I just want to pause. I just want to interject right there, because I wanted to just say, like, yeah, like biotech loves these viruses. Actually, the history of molecular biology also relied heavily on these viruses. That M13 phage, which is one of the filamentous phage that they study, that's how people made single-stranded DNA at some point in history, right? Like, people were using the replication of these viruses to create the molecules that maybe we take a bit for granted now because we have machines that make them, we have a PCR that makes them. Like, people were relying on filamentous phage to do many important tasks in molecular biology. And then that was extended at some point to work on this directed evolution. Actually, it's an evolutionary it's an evolutionary technique called phage display, where where each filamentous phage has a different copy of DNA in it. That means it's making like a different fusion protein attached to one of its coats, and then you can sort of like sift through all the phages to find a protein that binds to something, let's say, and then evolve it a little bit more and find the next best binder iteratively. So it's kind of wild to me, actually, in reading the introduction of this paper, to say we actually didn't understand a lot of basic biology around these phages Like, we knew how they replicate. We could make them grow in other bacteria, right? And we could sort of manipulate them in these evolution experiments. But we didn't actually know. We don't, and we still don't know, actually, I would say, after reading this paper. We still don't really know, right? Like, how does it even get in past, like, two membranes to get into the cell? Like, some idea of it, but, like, the level of detail, mechanistic detail is lacking. And, I mean, this paper, I think, pushes that forward by giving us this structure that then people can say, like, okay, it kind of fits here. It's kind of binding here. Like, these are some of the places that we would see these, how we think the mechanism is happening of this getting in.
Yeah. So, I mean, this is their, like, a model of their nanorod where the P7 is like what would have all the DNA trapped, and hanging off the bottom are essentially what are squid tentacles of like the attachment proteins to it. And and so I'm going to move away from this very phallic looking image to the the actual infection strategy that they propose, because they've got like two sets of proteins. There's a P3 on the outside, P4, P6 on the inside, and I think what happens is like P3 attaches to like the outer membrane, and then it opens up and injects the P6 into it. I think that's the impression and it's got this like cool little like hinge motion that where it just like opens out and pushes in the other like fusion apparatus to the other membrane on. So it's essentially trying to join up. So it's got these transmembrane domains, and then once it's like it binds to the cytoplasm, it kind of brings them together and then injects it. So again, half the time when I'm looking at papers, I look at the animations. I thought this was a cool animation.
Yeah, no, it's really interesting. Again, like, this is something that not knowing the structures of things, like, I think they must capture them in different stages, right?
Yeah.
Yeah, they capture these cryo-EM images at different stages, and they're like, okay, well, this is how things could move. They don't get the dynamics of it, right? Like, we only ever see snapshots of things. But by seeing the shape of them and then seeing the shapes before and after some sort of injection, like, yeah, you'd be able to sort of infer a little bit more that people didn't really know before this point.
Yeah. And
that's everything I want to say about this as well. Like, I mean, I think I love Cryo-EM just because it has given us access. It's interesting to hear that you were saying that, or that they were saying that, there's still limitations on the size of things that we could Cryo-EM, right? Like, in the world. At some level, I'm always like, yeah, Cryo-EM, you could find the structure of everything. And I guess that's true, but there's still finagling within the methods when you're trying to access, like, even more complicated structures. Maybe with, like, you want to see some before and after dynamics as well.
Yeah. So next paper is programmable protein delivery with a bacterial contractile injection system. So this is a paper where they're looking at about contractile injection systems. So we talked about a few of them. I think last paper
last week we talked about them as like they found them in some sort of soil bacteria as maybe some sort of salt resistance, something like that. Like, and then we talked about it like maybe a year ago, finding them in like algae thylakoid membranes as like maybe a way of like killing other cells, like as a defensive mechanism, maybe. Like, but yeah, like, it's kind of cool that we've talked about them in their natural state a bunch of times. Like, what might they be doing in nature? But, I think humans like to use the things that they've learned to try to make interesting tools. So, yeah, the authors of this paper are going in and thinking about them as, well, how might we use these things to maybe do something interesting for us?
Yeah. And these things never really struck me before, but they are like separate. So they are like produced and they hang out, like they're extracellular and they can deliver payloads. So they see in this case, the researchers were wondering like what can we actually fit into these payloads, and how well can we target them to specific cells. So they focus on one subtype of these from, I think, Photorhabdus, which is like a bacteria that forms associations with invertebrates. So photoreactive luminescence can make some worms glow, if I recall correctly. And yeah, they engineered it to hold certain payloads. So initially, the first one was like, oh, can it cause cell death? And then can it make cells glow? The two main things that microbiologists look for.
Well, they're nice. They're easy to look for phenotypes, right?
Yeah,
Stuff dying and stuff glowing. And I guess the other thing is that there are single proteins that achieve these phenotypes. And so they are looking for a payload, a simple payload to deliver. Makes sense that these would be the two that they ended up choosing.
Yeah. And the other thing they do is like they try to ensure that find ways to reprogram their target cells. So initially, I think these were targeted towards insect cells, and they find ways to target them to human cells by basically fusing them with antibody epitopes for some cells, or various.
On the tail fibers, they add things.
So the first thing they did was they did an adenovirus protein, and then they did some epidermal growth factor receptor proteins to try and see how well they can get these to adhere to human cells. And they found that they could use them to target the cells and even deliver things like Cas9. So they have cells that already have like the guide RNA in them, and then these will shoot into the Cas9 protein, and then that would end up creating in the models they use, I think they did like a Cryo-Electron model where they got it to fuse, and then that would produce an output. And and they even they tested out in mice as well. And
yeah, they end up injecting it into a mouse brain, and that's where the payload is Cree, and mice have the locked P sites. So if they're able, if these contractile systems can inject them, they'll do the recombination, and we'll see. I guess what do they have? Tomato, some expression of RFP.
Yeah, so I mean, a really interesting, because I think one of the big things is like finding ways to deliver proteins to cells now. There's so many different drugs that require proteins to be delivered. I mean, and trying to find new ways to deliver them in a good state into cells so they can activate, I think is really important. And also another thing that I want to draw your attention that I kind of glossed over accidentally was the specificity of cells. So the fact that they could attach these to antibody epitopes means that you can potentially target these to cancer cells, and even so this could be there's tons of possibilities, but that kind of specificity means that you can target specific cell types. If you're trying to do some kind of gene therapy type thing, or even just like deliver a drug to a specific cell, then these are really good for that.
Yeah, I mean, it's wild to think that, and maybe not so wild, because I was just talking about it when we were talking about filamentous phage, right? Like where you don't quite understand the biology of these objects, like how they're working in the natural, like how do they do their regular life? But already, with the knowledge that we do have of biology, right, we're still able to engineer and tweak the structures that we see to have them perform functions that are useful for us. And that is really, I think, the essence of biotechnology. And probably a good segue into the next paper.
Computational design of a dynamic receptor peptide signaling complexes applied to chemotaxis. So I think this is about engineering protein biosensors and then trying to figure out exactly how they're working. So I think this is the big part of this is actually the computation of how they do this. So there's a lot of computational modeling that's done as part of this paper to try and figure out.
Absolutely.
So they've got.
It's like Oh, sorry. Go ahead.
Sorry, no, you go for it.
I just want to say, so much of this paper seems to be the value of this paper is the work that they did on the computational side, figuring out how to predict a set of peptides that could bind in some, because proteins are really wiggly, and so it's not enough to the primary way that they've approached this problem of wiggly proteins is to sort of build things that kind of accommodate in some way that motion. And then it seems like they're doing like an iterative, almost like in silico evolution style, like algorithm to say, okay, well, then we don't want to have this wiggle, but we want to have this. They're trying to slowly move that that predicted binding to something that they have more evidence for, that is more stable, that like actually will transmit a signal. Because these are like big. They're working on essentially G protein complexes, right? And what I was reminded of when I was reading this paper was like we talked about like engineering G protein coupled receptors in yeast as like useful biosensors, because these G protein coupled receptors, they're really big and they bind to a lot of stuff, and it introduces like a really big conformational change across the whole protein. And I think that if you think about that, like if you imagine like a receptor as like a large mechanical object, right? A large clockwork object that when something goes on, it starts making motion within the whole set of the protein that ends up with, on the inside of the cell, a new shape that then can bind to something else. Yeah, essentially what they want to do in this paper is like engineer that whole motion machine just through sort of computational design parameters. I mean, they do experimental validation in this, but like, yeah, I feel like so much of the work of this paper is lost on me inside of this like this algorithmic approach to engineering G protein coupled receptors.
I mean, yeah, this is like that's the most interesting part of this, because usually with these sorts of papers, they do a bunch of iterations of things, which are essentially the equivalent of throwing stuff against the wall and seeing what works. But this paper, there is a concerted effort to understand why things work. So I think they had two design strategies that were quite interesting. So the first one was trying to basically map the peptide to all the parts of the binding pocket, and then find out which areas bind to it the best, and then upping that. So I think so to essentially focusing on the first shell of residues that are in contact with the peptide and then making them more bindy. And the second role was actually just to try and identify the binding and activation motifs that are compatible with the widest range of binding conformations. So not looking for the strongest, but the most diverse binder. And they found that like the ones that tend to be the most like they seem to find that they work very differently when you do those different design strategies. And my favorite thing about this was that they had these diagrams of the protein that kind of show the mechanical like motion of the protein, and how almost like a circuit diagram for how a protein works, where they show the thing that binds and then show like the direction of the movement that transduces the motion of binding into the actual receptor output.
Yeah, like I just want to return to that comment you said right at the beginning when you were speaking, like that most people would try, like if we think about these approaches, like when we think about screening through things, trying to find something that binds well, one way is like, yeah, you just try a bunch of combinations, you just see what works. But the paper here, as you were saying, like it is trying a bunch of things to see what works in silico, and by doing it in silico, they also are producing sort of like a mathematical representation of why they think those things work. Like what's the dependency of proteins that are deep, of residues, amino acids that are deep inside of this protein body that could be influencing the way that the signal is being transmitted from the outside to the inside.
Yeah,
I hope that makes sense.
I know that does make sense, and I think that's very important for if you're engineering proteins. This actually feels a lot like you're engineering them. That you understand how the algorithms are trying to understand how each part works, and then translating it to a way that we can understand, and so that we can
like not just looking for something that binds, and like just not looking for something that binds, and then sort of like using biology essentially, right, to like iterate and see like what. Just like go through so many iterations that we find the best molecule. They're really representing, I think, in code, like something about the mechanism. Yeah. Anyways, we're saying the same thing over and over again now.
Yeah, I think that's really neat.
It's really cool.
Yeah.
All right. Next paper we got here is microbial hitchhiking. or how Streptomyces spores are transported by motile soil bacteria. Yeah, so this is great. Actually, this is where I think Christoph joined us, because they had covered it on Small Things Considered.
Oh, right.
That ASM blog. And so I saw it on the Fediverse, and I was like, oh, it's cool. Like, part of doing YouTube, I think, is like we want things that like seem like they might intrigue people from the title and like from the technological perspective. And so this hitchhiking concept, I think, is just really awesome to think about. That there are so many bacteria, and in this case spores, out there that have no way of moving, and yet we see them distributed across large distances. And how did they do that? And of course, part of their moving is probably, they get blown and liquid dynamics and stuff. But there's also mobile microbes. And so maybe one of the ways that these objects move is by just aligning with something that's modal. And if they can hitchhike that ride, then they can get where they need to go. So even better, if those modal things are attracted to the same environments that these non-modal things want to live on, then you've got symbiosis made in heaven right there. And yeah, so this study starts with taking Streptomyces spores, putting them on a plate, and then having a bacillus sort of like swarm the entire plate, and seeing, do you get Streptomyces colonies that sort of form away from that initial inoculation point? And they do see that, which is quite fascinating.
Yeah, I love how they did one experiment where they put them both together and found like Streptomyces spores are spread around. But then they do things where they put like the bacillus like in a certain direction away, and you can see the direction that the Streptomyces spores are being swept. And that's
yes,
really neat.
Yeah, I love the picture. And I mean, I guess this appeals to a lot of microbiologists. Like, some of the early microbiology work is done sort of like in plate studies, right? Like the invention of agar essentially helps like set up these assays. Like, we've seen that like that antibiotic resistance one, which I also feel is like an evolution of like agar technology, just like increasing concentrations. You can see how the bacteria move. Yeah, I feel like this is like a really nice assay that they've set up to show that, yeah, the bacillus are moving. I think, they show that the bacillus moving towards plant roots, like in C.
Yeah.
All that junk is like, they did just like a Arabidopsis plant roots that they put on the plate, and showed that, like, yeah, they're moving. You could imagine that if bacillus is attracted to plant roots, let's say, they would be bringing those spores with them, and like really helping these Streptomyces, which well known to have symbiotic relationships with plant stuff, into that environment. And yeah, it's really what I liked about the finding here is that we're bridging this idea of like moving things in the scale of where these microbes live, right? Like between the scale of plant roots and the phycosphere or whatever, the rhizosphere, and just the bulk soil. Like, how do they even get there? It's not just feast or famine. It's there might be some tropism going on.
Yeah, and they even like,
what's this video?
Oh, this is a microscopy of Streptomyces being like attaching to the bacillus. And there are a couple of experiments they do deletions in the bacillus, right, where they don't delete the flagella, they delete the motor that spins it, just because like bacillus, they move in two different ways, right? They swarm and they do flagella, and I think they find that the what was it? Oh god, the Streptomyces, it doesn't need the flagella to move, but it does need them to exist in order to be dragged along for swarming motility. I think that's the takeaway from that.
That is the takeaway. Bacillus is a really well-studied organism. They have like a whole bunch of fun deletions that they can do. Oh yeah, that's a good diagram actually where they show it, right? Like either they can move and they don't have flagella, or they can't move and they do have flagella, right? They look at these combinations. Actually, it's not bad. And yeah, like they need the flagella to attach to these bacillus is the takeaway. I think, what else did I find interesting here? Oh yeah, they look deeper into this, right? That's a cryo-EM that you just
Yeah, that's
drew
it.
up.
So this is a crime EM like visualizing how the flagella are attached to by these spores.
Yeah, they
Uh,
didn't really have a clear takeaway from what I saw in the paper about this, just that
hmm,
it's attaching to the surface. But that gave them this clue to then also play around with the surface of these spores,
yeah,
to say, oh, maybe the surface is important in this case. And so I didn't know this, but spores have rod-like structures that sort of crisscross the outside of the spore, and they know the
yeah,
genetic basis of those rod. I think they're called rodlins. Is the protein family.
yeah. That's right, Rodlin.
And so if they delete the rodlins and you then get a smooth spore, essentially, then the flagella, then the spores can't bind to the flagella, and constantly they don't move along the surface. And I think I think that's the big takeaway, really, that needs flagella, needs a structure on the spore, and sort of the exact binding moiety they don't know, but they actually provide they provide a hypothesis that the spacing of the rodlins are kind of like
Mm.
grooves that the flagella perfectly fit into. So it could just be kind of like a Velcro-y type of scenario that
Yeah,
they get embedded
like the
into
birds.
grip tape.
Yeah, like literally, have you ever run if you ever run through like a field and been attached to by these like weird like spiky burrs that
Yeah,
were
yeah. This is
racing
maybe
through?
at the microscopic scale.
Yeah, so yeah,
Yeah, and so I just this was fascinating. I feel it definitely appealed a lot to my microbiologist, looking at plates and setting up a very interesting assay that sort of takes advantage of the fact that we're growing microbes in spatial environments, and then just looking at a macro scale, essentially. We didn't have to do microscopy to get that initial observation. Um, and that's something that I can really get behind.
yeah,
All
and I
right.
think I'd like to move on to the next one, which is uh,
Yes. Distinct glycoconjugate cell surface structures uh, make the pelagic diatom Thalassiosira rotula an attractive habitat for bacteria. So yeah, I guess sort of building on the theme of stuff binding to other stuff. This is a big, attachment is huge in the microbial world because things are wide, things are big, and bacteria are small, and they really need to be where they need to be close to what is going to make them happy. And so in this case,
yeah,
they're talking about one of the most abundant diatoms in the ocean is this Thalassiosira rotula. Um, they're algae that are covered in a silica shell, and then there's all these threads that are made of. Some are made of chitin, some are made of different glycoproteins. It's a whole mess of things, actually, quite complicated. But they do EM and these beautiful images of the algae, of the diatoms, encrusted with bacteria. Because bacteria, they like to live on the metabolites of algae. Actually, this is maybe a
yeah,
callback to we talked about some bacteria are opportunistic on algae, right? They like
yeah,
to kill the algae if the algae is weak enough to eat its nutrients, but they're also happy to live symbiotically and just live off the effluent that are coming off of these structures. Um,
yeah,
so they take these EM. This is in some ways just a big survey, a big microscopic survey of seeing that bacteria do associate with these diatoms, and then they grow up the diatoms actinically, meaning without any bacteria, so they're sterile
mm,
essentially, and then they reintroduce bacteria, and they see, oh, does that change the binding structure, the binding patterns? They also use a set of stains called lectins, which are sort of just molecules that bind carbohydrates. But these
mm,
lectins also have some fluorophores attached to them. And so when I guess part of the study is the resolution of the images that they're taking. Like with confocal microscopy, they're able to really precisely see, these algae have these specific lectins in specific places that bind to specific bacteria. And
yeah,
I think that's as much as I can say really about it, because the paper reads like a really long description of like every pairwise, right? Of like, oh, this lectin with this bacteria, not
Yeah.
this bacteria, but sometimes this lectin, like this bacteria in tufts and this bacteria only in like one punctate area. And it's like, you know, I can't really explain the complexity of that to you, but I'm sure for the people who are studying the bacterial between diatoms and their partners, this is like an incredibly useful atlas of things. And for us, some incredibly beautiful images of bacteria attaching to these algae.
Yeah. Yeah, I think it's mostly like these are the images are the main thing, and the rest of it describes what's going on, and
Yeah.
you're building a story from that. But yeah, I can understand how it's quite hard to sometimes scientific papers aren't written with a story in mind. Sometimes they are written to make it most convenient for scientists who are working in the field to pull out as much data as they need. So,
Mm-hmm.
uh,
Mm-hmm.
but
Yeah, like
yeah,
it's
this is
really like I imagine like if you studied one of these organisms and right one of these pairwise combinations, this would just be like a great like, oh yeah, I'm glad they can see this, and like now I know,
yeah,
right? Like I'm going to use this bacteria, I'm going to use these lectins, and I'm going to create a little model and try to say more about the biology here.
yeah. And those scientists would be really frustrated with us right now who are being so vague about it. And they're like, I want to know about this specific bacteria. Can you tell us? I'm like, oh, whoops. Sorry, just read the
Yeah,
paper.
yeah, exactly. Got to read it yourself. I'm sorry.
I'm sorry. I can pronounce maybe half of the names,
Yeah.
but
I think like one of the things that what I learned from reading the paper, you know, everyone picks up something different when you're reading this big text of this wall of text. I didn't realize that like there are pores inside of the diatom shell, and like they could see specific bacteria like associating around those pores, because that's
yeah.
where the nutrients come out of, right? Like that's where the you know the algae inside is doing some metabolism, and like all those metabolites are streaming out of these waste pores, and so bacteria, you know, some bacteria preferentially attach to those zones. And it's just great to have that image of like all this complexity happening at the tiny scale, right? To understand that like distance does matter, and you know this is why I think it's a nice transition to like from the last paper into the final one that we're going to have for you. But like yeah, like really imagining the micro scale and how much distance has to be covered by some of these organisms in order to get to where they really need to live.
Yeah. And
Um,
I guess staying in the sea, but in a similar topic, got chitin utilization by marine picocyanobacteria and the evolution of a planktonic lifestyle.
So actually, this is like going from a very abundant algae, a diatom that's out there, and moving into a very abundant cyanobacteria that lives out there in the water. And so Synectococcus, I think SAR11 is part of this family. Essentially, what they're curious about is do these like there's a lot of chitin in the ocean. I think I mentioned in the introduction molecule that's found in arthropod shells and zooplankton shells, right? It's a yeah, and in that previous paper, diatoms also have a bunch of chitin in them. So this is a very abundant molecule, and people are curious to know whether this extremely abundant bacterial group can metabolize any of that chitin. And so they go in bioinformatically and they look for potential genes that could do this chitin metabolism. And I think what's interesting is there's some interplay between chitin utilization and peptidoglycan recycling. So they look for both those classes of genes in order to build a list, a putative list of like where that metabolism could be happening. And they do find it in a bunch of different clades, even though you wouldn't think it, because these are cyanobacteria. They should just be eating. They should just be using light to survive, and yet they have this machinery to digest chitin. And so that's actually really curious for these scientists. And so they sort of follow up a little bit on that, and they show that the bacteria use chitin only when they don't have light. And so it's basically a sort of a secondary metabolism that they have, right? They can switch into using chitin as a carbon source if needed. Yeah, and they can attach still. I think this picture that you're showing here is that they're attaching to chitin. So that's an ability that they have, and they weave that into an interesting story of evolution, actually. So maybe moving all the way to the last big year.
Yeah. Yeah. Yeah.
Figure 7.
Yeah. I think.
Yeah,
Yeah.
they weave that story, like they go, so like why, right? Why do these cyanobacteria have the ability to attach to chitin and also digest it when they are free living bacteria in the middle of the ocean that use photosynthesis? And so
Mm.
the theory, and I think it is sort of motivated by seeing the like where these abilities fall within the evolutionary history of all the Synectococcus. They say that, oh, maybe it's an ancestral way. They used to be associated with chitin, and they used to be like just organisms that lived off the detritus. But then at some point, they were carried out into the middle of the ocean on rafts of these tiny particles, and then out there in the open ocean, they could just they could specialize into just being just requiring the light of the sun, and they wouldn't have as much access to chitin out there anyways, right? And so that's the story that kind of gets built up by these observations.
Yeah, it's quite interesting how they go with this, because basically they're like they do some interesting like molecular clockwork to try and figure out, okay, so these chitin genes evolved around 570 to 420 millennia years ago, and that's about the same time where chitin evolved in arthropods. Coincidence? I think not. And I do like understand like because the idea is that these started off as like microbial maps that lived on like the sea floor, and that was
right.
the
That's
only place
the
where
beginning. That's the beginning of microbes on planet Earth, right? Was that in the shallows there was like these big. Well, first light could reach the shallows, and then it was advantageous to live in these colonial map forms. There's fossils of evidence of this sort of stuff.
Yeah. And the idea is that there are like various like was it archaea that were living the rest of the place. So there's this theory that we used to have like purple oceans, and like the green bacteria, the green like the ancestral plants lived like at the bottom, and where they got the green light, and all the other light was chewed up by like these purple bacteria that are out there, and that this kind of indicates a way that these bacteria went out and started to colonize the rest of the ocean by learning how to be more free living. And essentially, so
by catching a ride on these, right? Like by essentially like bootstrapping their ability to attach to chitin and use that chitin, and then take that into a zone where then they could access. They could just live by themselves. So have a new niche, right? I think they say that here, niche partition.
Chitin was like the Dumbo's feather that they needed to learn that they could fly on their own.
And maybe one last thing I'll say, since we did a shout out to Small Things Considered, which is an amazing blog that you guys should go read. Like this also reminds me of PBS Eons, which is another great YouTube channel out there. That just like it's interesting to hear like what stories we can pull out of the past just based on these like scraps of molecular information, right? That get pulled into like a really interesting story about how things might have been. So
I mean, always very interested in. I mean, we'd had that bacterial fossils paper a while ago.
It's
very interesting.
In the
like,
chert, right?
The Rhiney chert paper.
Rhiney chert.
Because like bacteria are like the most a very primitive form of life, and it's like almost like for life trying out everything that could be tried out, they try out in bacteria, which is why they're so fascinating. You get the craziest stuff coming out, and that's that you wouldn't see like magnetotactic bacteria or like bacteria that live inside other bacteria. They're also or like I'm not even getting to viruses. There's so much like interesting biology and interesting stories here that the phages are fighting each other using bacteria as a battleground. Tons of fun.
And we've talked about it on this channel, right? Like there's a huge backlog. It might be a bit hard to get through. I mean, at this point, I would then recommend like looking through our Zotero for some of these keywords. Like you might find those papers, and I've tried to, at least in recent history, I've tried to put those papers then attached to the to the specific episodes that we've done. I'm sure you could also search them in the episode list or in YouTube maybe.
Yeah.
We haven't tried that. Like, my recommendation is full text search in Zotero, then find that paper and find the episodes associated with. But you may also try to search as well. That would give you the titles, because the titles are always in our descriptions.
Yeah. Also, like I know that there's like a deep dive playlist that I've created, but I haven't updated in a while. That could be a good start, because of the news articles we pull out a bunch of things that we are interested in, and it might be a lot. And a deep dive, you can look through and see if there's something you're particularly interested in.
Yeah.
Then you can go and figure out more about
that. Or also, if you see something that we've covered in one of these past episodes, you could tweet us or toot us or leave a comment saying that you want us to dive into it a bit more, and we'll try to do a longer sort of episode.
Yeah.
So, and all that being said, join us in 2 more weeks where we'll be doing this again, covering a bunch of cool microbiology papers that we've seen.
Yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter or Mastodon. We both believe that peer review is a process, and so it requires folks to have read the papers to make it work. If you think you have something to add or found something unclear, let us know in the comments.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content.
2 weeks.
Until then, goodbye.
Bye-bye.